Hi, I'm Dan Fantastic Roaster, and on this week's episode of Urban Legend Destroyer, we're going to be talking about phylogenetics and ctenophora. Let's get into it. So this week we're going to be looking at ctenophora and all their fun, interesting biological facts, and look at their phylogenetic trees in relation to porifera and how they relate to one another. Let's get into it. So ctenophora are hermaphroditic, carnivorous, ocean-dwelling organisms, and there's about 100 to 150 species within the family, and they have a bioluminescent quality to them, and the color's generally green or blue. The ctenophora have a life history that starts with unilateral cleavage, sidipid larval stage, and then plankton. And then there's the porifera, which is just a sea sponge. Porifera are generally round and tend towards a cylindrical type shape, but are more uneven while being attached to rock or some surface in the ocean. And they're not a free-floating organism like the ctenophora. But based on their similar shape, it is not unreasonable to create an idea that there's a close evolutionary link between ctenophora and porifera. But how is that true, though? How closely related are these two organisms? Before we get into that urban legend, here's some background information about porifera. Porifera are oceanic organisms that can reproduce sexually or asexually, obtain nutrients from food based on a filter feeding system. But let's take a closer look at each of these organisms. So here's some more in-depth information about ctenophora that we will then compare to porifera and see how closely related they are on the surface. Ctenophora have appendages in the form of tentacles so that it can hunt its prey and move about the water, and it has no segmentation. And its support system is a hydrostatic skeleton, and using its appendages and hydrostatic skeleton, it can move through the water with an undulation-type motion. It gets oxygen through respiration from its ability to diffuse across its skin layers, which are 2 to 3 layers of tissue. The ctenophora have no circulatory system, though, and its digestive system has one opening and is both intra- and extracellular. Ctenophora have no cephalous action, which means that there's no central head to move around, so it only uses its body to move about. And it has an asolomate body cavity, which means that there is no body cavity. And it has 2 to 3 layers of tissues with biradial symmetry, and biradial symmetry just means that it's both bilateral and radial symmetry. So they're ctenophora round, about a central axis in shape, and so one part of the organism is identical across the central axis, but not on the adjacent side. Now, porifera, on the other hand, have a few different traits. They have a few similarity to our ctenophora friends, but they also have a lot of differences. First, that major difference is that porifera have no nervous system at all, which kind of makes it seem like there is no tight connection between these two organisms, but it could be different than we expect on our phylogenetic tree, so let's get into the rest of the traits. Next, porifera have no appendages, segmentation, or form of locomotion, as this organism is attached to some underwater object or structure, such as a rock. It does have an endoskeleton, which is a bit different than the hydrostatic skeleton we see in ctenophora, and it respires oxygen through small pores on its exterior skin. Another similarity between porifera and ctenophora, which also respire across skin layers. Both organisms lack a circulatory system, which adds to the evidence that there is some sort of close evolutionary link between these two organisms. Porifera digest through a system with 2 openings and an intracellular setup. Porifera, however, have the same body cavity as ctenophora with an asolomate or lack of body cavity. And both don't have any cephalous action, and they only move their bodies about, which adds another shared trait between the two oceanic organisms. However, porifera have no different layers of tissues or symmetry in their organization. Two organisms visually, and from dissections in previous studies, we can find that there's a lot of, similarities and differences. But the biggest differences between these two organisms are that ctenophora have a nervous system and multiple layers of tissues. Before we get into the results we obtained, let's take a look at some published finding about ctenophores. The first being from Wahlberg et al. And the second being from Tang et al. They found that ctenophores are suspected to have originated somewhere between 505 and 520 million years ago, and that ctenophores were among the first Eumetazoan lineages. Now, let's take a look at their phylogenetic trees and information before we get into what we found. It is known that ctenophores did not originate by their symmetry. This clade does not seem to be given a name, so we will refer to it as the inclusive group, including Cnidaria and Bilateral, but excluding ctenophora as Planulosa. Possible synapomorphies of Planulosa are the presence of endodermal myoepithelial musculature, the septate junctions joining the epithelial cells, among other traits. The uncertain placement of the organism on the tree is due to a multitude of conflicting interrelationships that have been inferred, causing discrepancies among different trees. Without supporting hypotheses regarding the closest relatives of bilateria, animals with bilateral symmetry, there is no clear path to determine the transition to a worm-like body with a mesoderm and sensory and nervous system at the anterior end of the organism. Their trees look different from ours, however, because the article looked mainly at the SSU rRNA sequence of the organisms to come up with their tree, and we use 16S rRNA and the characteristics from our data matrix. So there are two phylogenetic trees we here at Urban Legend Destroyers made. The first being based off the data matrix of information and characteristics we discussed earlier, and the second being a 16S rRNA tree. Let's get into it. This first phylogenetic tree is based off the characteristics we discussed earlier and assembled in a data matrix. We found that the tree based on this data matrix found the relation between ctenophora and porifera were not as closely linked as we thought, as we found that ctenophora are in between echinodermata and annelids, with ctenophora being separated by porifera by two layers, being cnidaria and echinodermata. However, if we look at the 16S phylogenetic tree, which is based off of sequenced rRNA between the organisms, we find that porifera and ctenophora are closely related, being right next to each other. In fact, it goes, as we can see here, porifera, ctenophora, and then cnidaria, which is a bit different. And this is based off of rRNA, not any visual inspection. This indicates a much more specific and accurate examination of these two organisms, because we are no longer visually inspecting, but using the structures and processes of the organisms to define them. These differences could be due to the fact that they are more closely linked genetically than their physical and visual functions would indicate, as they look similar but with a few key evolutionary updates. This makes sense because porifera are some of the most basic and simple organisms that were under investigation, whereas ctenophora are slightly more complex, albeit still very simple compared to other organisms like arthropoda or mollusca. So, we're going to confirm and not destroy this urban legend.